we are tired. I'm going to be short this talk. Thank you for the invitation, Lorena and Maribel. And this work was done in my previous postdoc position at the Metropolitan University and in collaboration with the Human Science Center when I did my PhD. So, this project was titled Phenotypic and Genomic Characterization of two heavy metal and temperature tolerance Cupriavidus gilardii strains isolated from the Naica mine. So already this work has been published for more details you can see on PubMed or something like that. Well, the Naica mine is a mystical place because it's heavily the biggest deep crystal in the world, reaching up to 10 meters wide to 100 width. These crystals are dating for nearly 400,000 years ago, and the scale was fully covered with wire. It was in the past, obviously, and this place is show annually temperature of 55 degrees, and over the year, and near it to 100% of humidity. So, highlight this place is like this Naica mine. It was very important source of lead, zinc, and copper and silver in production in Mexico since late 18th century. So, in this picture, we observe the Naica underground system. In the yellow line, we observe that the caves and mine shafts. And it is after 700 meters depth. And we observe 1,000 meters intrusive magmatic body that this favors the temporal condition of the cave. The Naica mines comprises several caves, like crystal caves, phenolic caves, and González cave, and others, but these caves are connected by tunnels. These caves are located at 219 meters depth, and it is interesting because each cave has different kind of crystals and different sizes. So, the research of this mine is curiosity because only had was this, researching up to early 2000 years, and this in online the slide we observed the principal discovery that was done. Several research like geologists, mineralogists, astrobiologists has involved in this discovery. But in microbial research, is a little bit information about this. But in several, in Mexico, we have a little bacterial isolate from this case. This collection is belonged to CD and Hercules, and we observed that some of these bacterial isolation. But in this talk, we only focus on Cupriavidus isolates, the like this, and these isolates were from iron oxide deposit. For example, from the Kendall's cave clone, as in the picture see. Previously in this work, these isolates were classified into Cupriavidus gilardii genus and the Taiwanese species. So this genus of this bacteria has been described as very potential in biotechnological application. So like herbicide degraders, growing in extreme environments, high tolerance to heavy metals, nitrogen fixer in the leguminous plants, and others. Given these approaches have been reported, we select only these isolates, and previous analysis will observe that these isolates were positive to for specific tests, like this picture. and is correlated with the central point that we isolated. So we only sequenced two Cupriavidus isolates, and we did a genomic comparative genomic analysis of these clones. So this slide we observed the summary of genome features, and we observed that we use a path barrier and using a platform for sequencing of genome of these strains. We observed that the features are similar between strains, NUB and OB strains, but not only in the isolate were similar. The number of CDRs, number of chromosomes, and others genome features are similar with other Cupriavidus species. With these results, with the genomic data, we identify 510 genes across genes, and we constructed a phylogenetic approach on this picture. Also, we constructed a branch nucleotide identity analysis, and in both cases, we observed that the isolates of Naica were grouped onto cluster gilardii species. Given that this approach, we reclassified with this isolate because previous was Cupriavidus Taiwanensis classified. Only in this analysis was 69 RNA sequences, and we did a robust taxonomic classification. So also we did phenotypic characterization of this strain, and we observed in this table the carbon source assimilability test, and we observed that the Cupriavidus isolates are here, and other Cupriavidus reference strains are included. The Cupriavidus mecata, Metallidurans, and Taiwanese. And we saw that these all strains can be grown on this carbon source, and only with, for the current situation. But in some of Cupriavidus can grow with malanato, lactato, and malato. And these results are correlated with the genes in. Because, these results are correlated with the absence of the genes involved in the glycolysis and pentose pathways. And there is only a Cupriavidus gilardii strains are absent some genes involved in this pathway. These results show that could be these strains may possess highly specialized metabolic pathways. It is, of course, more experimental. Well, also we identified the range of temperatures of these strains. Also, we included a Cupriavidus reference strain. And we observed that only Cupriavidus gilardii species can be grown up to 48 degrees, and in contrast, the other species can be grown in these temperature conditions. Also, we determined the maximum specific growth of this isolate strain that was 42 and 45 degrees. This result suggests that the bacteria are thermotolerant bacteria, and it's a specific characteristic of this isolate from the sample size. Also, given the previous results with the ferric precipitation test, we only focused on the heavy metals tolerance, and we did experiments with the heavy metal resistance, and we included the most studied strain that was Cupriavidus metallidurans. is other basic research model in this topic, and as a control, obviously, another Cupriavidus gilardii as a reference strains. So in this condition with copper, we observed that OB strain were most tolerant to copper than NUB strain, sorry. But in contrast, the NUB strain were more tolerant to cadmium and zinc than Cupriavidus OB strain. Also, we observed that the Cupriavidus gilardii CCG strains reference were more tolerant to nickel than Cupriavidus gilardii isolates from Naica. This result suggests that the heavy metal clusters or mechanisms are involved in this tolerance. So we search all heavy metal resistance clusters identified in Cupriavidus metallurans, and we search in their genomes of these isolates. We observe that these clusters are encoded with the heavy metal resistance for Cupriavidus metallurans, and we observe the presence and absorption in other strains. And we observe that of these strains lacks the CC cluster. These CC clusters are involved in cobalt, zinc, and cadmium. Also, the MF cluster involving zinc and zinc leads cadmium and cobalt resistance are absent in Cupriavidus gilardii CCG strain. So, it's interesting because the CC cluster was the most interesting cluster because some genes involved in horizontal linear transfer are flanked to this cluster on Cupriavidus metallurans. But in other Cupriavidus gilardii, we observe that the same sampling of the genomic context. But in other Cupriavidus gilardii could be like other genes. So this result suggests that this cluster could be acquired by linear transfer in Cupriavidus genome, not in Cupriavidus gilardii. Finally, this is our conclusions. It was a short research of this topic, but I think it's interesting. It responds demonstrate to the approach genetic combination with the phenotypic characterization. This study clarified the taxonomic classification, for two novel Cupriavidus gilardii strains, and the well-known sequencing data using bioinformatic analysis we identified 510 biomarkers suitable for taxonomic classification for Cupriavidus genes, and the sensitivity to heavy metal resistance we observed that is correlated with the presence and acceptance of heavy metal resistant clusters in the genome. Obviously, the CC cluster is interesting for more study because could be this cluster was acquired for horizontal genes and could be the first study with this heavy metal resistance. And this event suggests as a vital mechanism for spreading heavy metal resistant capabilities within a moderate Cupriavidus population. Obviously, this project was important about biochemical characterization, and these studies could be involved in the future for bioremediation or in contaminant cells at high temperature. And finally, I want to thank the all people involved in this project, my past advisor, BiGEvo, my past professor and my past advisor from the PhD studies, my colleague Luis Lozano from CCT, and obviously Mariana, my wife, and obviously all institutions that have been working with. Thank you for your attention.
There's definitely time for questions. Somebody? Lunchtime was so long ago.
Thank you for your talk. I want to make a crazy question, as somebody says before. Seeing that your microorganisms are tolerant to heavy metals, do you think that they could possibly have a role on mineral formation in this site? Do you think that we are looking at organisms that could have formed the mineral associations in the Naica mine? Or do you think that they are only tolerant because they are in this environment?
No, for answering this question, I don't know the information about heavy metal tolerance because this study is the most studied in this topic. And has been reported that this strain has ability of bioaccumulation and biotransformation of alcohol ions. So if we separate these residues with these strains, we could be formulating hypotheses from this role, from these strains. And the answer could be, say, these strains could be stayed here for the effect of toxicity of heavy metals and the transform to other species ion, not than reactive. Yes, for me.
Yes.
Thank you. Dr. Gavati, so my question is, you suggested that that whole operon or sets of genes are flanked by transposons. So are you suggesting that that set of genes gives the Cupriavidus copper resistance? And if so, can you just take that region and transfer it, say, to coli, and do they become resistant?
Yes, in this project, it doesn't have this opportunity to do because it was short, but in several studies have been done in heavy metal elements. The expressions of the organism in this cluster can be acquired the resistance of heavy metals. Could be possible to do in these strains for past this whole cluster. But the first question that I did is which could be not in total completed because the fragment is to be large. And could be I don't know why plasmid or what, but the vector could be safe as introducing E. coli with all cluster.
Just a quick follow-up. You showed right at the very beginning, that this organism has two pretty large chromosomes.
Mm-hmm.
Right?
Yes.
Is there anything interesting about which genes are on which of these chromosomes?
Yes. In the paper we discussed the harboring in second chromosome, the principal genes for physiology like division cellular and others. And we found some genes involved in the replication like a plasmid. And this discussion of the second chromosome could be called chromic, not chromosome, but the spread of the genes, essential genes. And could be in this case, more studies are completely needed for analysis.
salt concentration is best for your isolate?
Concentration about what?
Salt concentration. Have you tested for allophilia or allotolerance?
And these strains I don't know a lot of them, because they like chloride sodium, which 1% died, and they are in a lot of them.
They don't live
No.
above 1% sodium chloride.
Yeah.
And pH, do you know?
Up to can go 9 to 10.
One more question.
Here.
Thank you for your presentation. I just find curious that your isolates from this case that is regularly at 56
55.
65 degrees, and you tested for the thermal tolerance, but they don't grow above 50 degrees. So why do you think that happens?
Yeah, it's a good question because, as you know, the bacterial community has other species of bacteria, and could be these other bacteria that are most tolerant that this can be protected. So, as you know, also in this ecosystem have macro ecosystem. And so this could be these two probably